Welcome to our CaTS COVID-19 Chronicle seminar series. These have been really outstanding so far, and I think we've got a great lineup this afternoon to keep everybody informed on what's happening about COVID. So our speakers today are Dr. Miele, Dr. Cormier. I'll be very brief in my introduction with Dr. Miele. I could spend probably 45 minutes on all of your accomplishments, Lucio. So pardon the brevity in that I want to make sure we got enough time for both speakers. And sometimes, you know, we'll have to be sensitive to that, 30 minutes each, and make sure that we can get through this. So I don't want to eat it into your time too much, but I do want to point out to everybody that during the talk, if you have questions, put them in the chat window, and we'll save a little bit of time at the end of each talk to share your questions with the speaker. So without further ado, Dr. Miele is coming to us from LSU Health Science Center in New Orleans. His medical training was in Italy. Lucio is a genuine 100% Italian. And his graduate training was at the Max Planck Institute for Molecular Genetics in Berlin. He went on to do a fellowship at NIH. He worked with the FDA as a senior investigator on monoclonal antibodies, which is very pertinent today. And he eventually became acting chief of the Laboratory of Cell Biology. He's worked at positions at Loyola and the University of Illinois, Chicago. Followed by a stint at the UMMC Center in Jackson, Mississippi, where he was director of the Cancer Center there and Ergon Professor of Medicine. So we're very fortunate, or I should say, Dr. Nelson was very fortunate to recruit Lucio to New Orleans. He is professor and department head of the Department of Genetics at the LSU School of Medicine. He's director of the interinstitutional programs at the Stanley Scott Cancer Center and the LCRC. He's also the Cancer Crusaders Endowed Professor in Cancer Research. For CaTS, he serves as director of the CaTS Biomedical Informatics Core, and he's been really a main contributor for our N3C initiatives. As you might expect, Lucio is a very prolific scientist. He's authored over 245 publications. He's sat on multiple NIH study panels, grant review panels across the board, NIH, NCI, NCATS, and DOD, and has been just an outstanding contributor to science and also a great leader here in Louisiana. So thank you, Lucio, and the floor is yours.
Thank you so much for this very kind introduction. Let me share my screen. And go to, here you go. Everybody should be able to see this. Okay, so today's talk is a pragmatic approach to testing, or how to make lemonade. The subtitle will, the reason for it will become clear in a second. As we all know, the miracle didn't happen. And these two graphs were updated yesterday. The incidence and mortality of COVID-19 in Louisiana and the United States and the world has kept rising. Almost 1 in 10 people in the state of Louisiana have been infected with SARS-CoV-2, and approximately 1 in 58 have died of it. Right now, again, this is as of yesterday, the distribution of the virus has moved to the most rural parts of the state, but it remains quite abundant in Baton Rouge as well as in New Orleans. So the problem has not gone away, and it's not going to go away anytime soon. What I'm going to talk to you about is what we at LSU Health Sciences Center New Orleans did when we started seeing the first wave of COVID-19 and how that has evolved in time and what we have learned from our experience. So what we had at the end of April was a 6,000 employee campus, many of whom are involved in patient care, so with significant exposure to patients and to the public. Students, residents, and faculty who visit multiple healthcare facilities in Louisiana, not only in New Orleans, but also in Baton Rouge and in other parts of the state. And major supply chain problems with critical shortages of just about everything: swabs, medium, PPE, test kits, plasticware. Let's just say that no one was prepared for this, and we were no exception. We also had no dedicated personnel for testing, no dedicated budget for testing, no dedicated space for testing, and we tried for months to resolve complex negotiations surrounding the use of symptom reporting apps and exposure notification apps. The state of Louisiana finally did start using one last week. What assets did we have? Well, we had some significant expertise in both molecular testing and immunology. We had a lot of committed volunteers from multiple schools on campus and multiple departments within these schools. We had a $100,000 donation from Hyundai, who basically bought us some kits, some testing kits from South Korea. And we had some clinical space, unused clinical space within the LSU Healthcare Network primary outpatient clinic, the Seton building. So we formed a committee that included epidemiologists, infectious disease specialists, and a number of other people, including myself. And we came up with what would have been an ideal containment plan, also following CDC guidelines and other epidemiologists in major institutions. Of course, mandatory masks and social distancing, work from home whenever possible, and lockdown if needed. Universal use of a symptom reporting app, such as COVID Check. COVID Check is the one that we ended up using, but it wasn't the only one in the running. And ideally, an exposure notification app using either Bluetooth or GPS technology, repeated testing of everyone coming to campus with a highly sensitive and specific molecular test, rapid turnaround time of test results, quarantine, and contact tracing of positive cases. I don't believe anyone in the country was able to put together this kind of containment plan in a short period of time. And we were faced with a number of difficulties. So instead of doing this, we were handed a lemon and we had to make lemonades. So this is what we actually did. And the plan evolved over time. We did have, of course, mandatory masks and social distancing, and work from home was allowed whenever possible. We never went into a full lockdown, unlike other academic institutions. We didn't have enough kits. And so we limited PCR testing for symptomatic individuals or patients with high-risk exposures. And for that, we used an FDA-authorized, highly sensitive 3-gene test, the C-gene Allplex. This was not one of the most well-known in the United States at the time because other companies were getting a lot of publicity. But this is the test that South Korea used to crash their curve. At the time when we adopted it, thanks to the Hyundai donation, it had been used in over 1 million people, and it has performed extremely well in our hands. It's almost too sensitive, if you will, and it's highly specific. We also put into place exposure surveillance with a rapid antibody test that could tell us within minutes whether somebody had been exposed, specific for asymptomatic individuals. And we added a twist to this, which has turned out to be quite helpful. At least it did turn out to be helpful at the time. We reflexively tested by PCR anybody who tested antibody positive. This encountered some resistance because a number of individuals on campus, particularly individuals with clinical duties, insisted that since they had antibodies, they had to be immune, and therefore they were not capable of transmitting the virus to anyone, and therefore they should go to clinic. As I'll show you in a second, that's actually not true. Our IT department set up an ad hoc registration portal for testing, which required symptom and exposure screening. Now, since the negotiations with COVID-sick were taking forever, what we ended up doing was creating our own questionnaire, which is similar but not identical to that of COVID-sick. We add the GI symptoms to that and a few other things. And so if somebody wanted to register for testing, they had to go through this questionnaire first, therefore giving us information on their symptoms. And if they were symptomatic, they were directed to being PCR tested and isolated, initially at the hospital and subsequently in the health network. If they were asymptomatic and had been asymptomatic for at least 14 days, they were directed to antibody testing. In addition to that, IT put together a self-reporting portal. Of course, since this was voluntary, it was supplemented by additional measures. We tasked department heads and other supervisors, for example, in facilities and maintenance, with reporting the numbers of cases in their departments to the Vice Chancellor's office. We had to go for something cheap because, as I said, we had no specific budget for this, and fast. So we put into place a system to report the results of antibody tests within 15 minutes and call back for PCR anybody who was antibody positive. What actually ended up happening in reality is that most people remained in the waiting room, which was socially distanced, until they heard back from their antibody result, and if they were positive, they moved on to PCR testing. The results of all testing, antibody and PCR, was made available through a secure portal. Essentially, what would happen is anybody would get an email saying your results are available, sign into the portal. Of course, PCR results were reported to OPH and to the School of Public Health for contact tracing purposes within 48 hours. There was a central number to call for information, which is now tasked to a campus clinic. This happened much later. Tasked to a campus clinic staffed with 2 physicians, a physician's assistant, and 2 RNs. Just to give you an idea, the original central number to call for information was mine. And I answered a large number of phone calls, and I ended up having to train my assistants to Q&A, because otherwise I would spend all of my time on the phone. So this was the actual workflow of that phase of our testing. Registration through this portal, which was accompanied by symptom screening. Symptomatic individuals were directed to isolation, PCR testing, and if positive, NOPH reporting. And then only after 14 days, they were allowed back into work. Asymptomatic people were directed to antibody testing. At the site of antibody testing, they were again screened for temperature and current symptoms. And also they were handed a questionnaire on past exposure and past symptoms. This turned out to be quite interesting from an epidemiology standpoint, and a good teaching device for our medical students, many of whom volunteered in the testing site. There was one particular case that allowed me to show one of our students how to take a history. The individual in question, when asked whether or not he'd ever been exposed or symptomatic, said absolutely not. Upon more accurate questioning, he revealed that both his wife and his child had been quite sick with it. So he obviously had been exposed. So there was a teaching opportunity there too. Antibody negative and asymptomatic individuals were allowed back to work, whereas antibody positive received immediate PCR testing, and if positive, isolation, OPH reporting, and again, after quarantine, back to work. This process was repeated as needed, back to registration, and all this information ended up in a de-identified data set, which we could do nothing with at the time, because this was not a research project. It was a public health survey. So this was not research. We thought that at some point we may want to use the results for research, which we're going to be doing now, generating 3 sources of data: demographics and location from the registration database, antibody results, IgG and IgM, as well as PCR results, including cycle number for an estimate of viral load. What was our experience? So I'm going to give you highlights because an in-depth analysis of the de-identified data from this is now ongoing under an IRB-approved protocol, but I can tell you overall results. Between June and December of 2020, we tested over 1,600 individuals, many of whom were tested repeatedly. Campus exposure rates, as determined by antibody positivity rates, were consistent but stable and high. On average, 1 in 5 on our campus has been exposed to SARS-CoV-2. On the last day of antibody testing, it stood at 21%, but it oscillated between 20 and 21% throughout the first 6 months of the pandemic. Interestingly, 30% of antibody positive individuals were also virus positive. So at population level, PCR positivity rate was generally stable at 6%. It's now a little higher at 9%, and that's because we're in a 3rd wave. But during this particular program, it stayed constant at 6%. That meant that gating asymptomatic individuals by antibody testing significantly enriched for PCR positivity. That tells you that 1 in 3 of the people who thought they were safe because they had antibodies were actually potentially infectious. And we had to drag a few of them away from operating rooms and clinics where they could have potentially infected other people. As reported by others in the literature, both IgG and IgM antibodies against the nucleocapsid antigen tended to appear simultaneously. We can discuss why this might be later, but it's not typical. We didn't see the typical IgM first followed by IgG. What we did observe, which is interesting from an epidemiological standpoint, were mini clusters of asymptomatic case. So we would see a positive individual within a unit, for example, a group of laboratories or a group of offices. A few days later, a number of people working in the same environment would test positive. However, because of the mandatory masks and social distancing, all of these cases were asymptomatic with low virus loads as estimated by CT values. What this is telling us is that asymptomatic transmission was actually common, but not pharmacological interventions, that is, social distancing, masks, hand washing, and all that, did limit viral loads. So there is a circulation of this virus that moves under the radar, and a lot of people who were not sick were actually infected. And thanks to masks, the number of symptomatic cases was actually quite low. Antibody responses did fade within approximately 3 months, also as described in the literature. We've seen several cases of 2nd and 3rd infections of individuals who had been antibody and virus positive months after the original infections. And there has been not a lot of rhyme or reason as to which infection was symptomatic. We had people who were symptomatic one time and then became infected again with asymptomatic cases 3 or 4 months later. But we also saw the opposite. We saw people who were asymptomatic, who clearly had not developed protective immunity because they became positive again months later, this time symptomatic. Or they may have been infected with a different variant of the virus. And this is something that we're going to get into in a second. Now, clearly, this was an emergency program, and it has evolved significantly. What we are doing now, we have retained the all-plex PCR test specifically for clinical purposes, that is in patients who are symptomatic or high-risk exposures. And our guidelines are that nobody should show up for testing within hours of an exposure because that's most likely pointless. We were letting people wait 3 days under isolation. Because this quick test has a quick turnaround time, and we're now using the electronic health record of the LSU Health Network for registration and reporting. We've centralized the dedicated campus health clinic, which is in the same building as the testing clinic. And thank God they answered the phone instead of me answering the phone. So we're now directing everyone to call that number. And we've set up an additional level of testing, which is based on next generation sequencing as opposed to PCR. This is the Illumina COVID-seq, which is an FDA authorized assay that can provide whole sequence of the viral genome in 384 samples at a time with a 3-day turnaround time. And we're now capable of high throughput variant calling. We are doing vaccination as a closed pod with the Pfizer vaccine. We've administered approximately 1,500 in a week. That will be followed by surveillance screening using the COVID-seq assay, which will allow us molecular epidemiology. We're validating saliva and nasal sampling for surveillance screening, so we don't have to NP swab everyone. And many thanks to Stephania Cormier for sharing her saliva protocol and saliva tubes, because those were also not available. And we've discontinued routine antibody testing because at this stage, it's no longer informative. Speaking of variants, nobody ever bet against Charlie Darwin. I have been saying this for months. The idea that this virus would not evolve is highly implausible, let me put it this way. And in fact, the virus has evolved and continues to do so. This is a portrait of the genome of SARS-CoV-2. It's an extremely compact and actually rather elegant from a molecular standpoint nanomachine. It's approximately 30 kilobases. This open reading frame 1 and B encode for the RNA-dependent RNA polymerase. This is the famous spike gene that encodes the protein that the virus uses to attach to its target cells. Here's the envelope, which is the most conserved among coronaviruses, the nucleocapsid, the membrane, and other accessory proteins. Not a single nucleotide is wasted in this thing. The degree of homology between the consensus genome of SARS-CoV-2 and SARS, the original SARS, is 82%. So it doesn't take a lot of nucleotide sequence changes to completely change the infectiousness and pathogenicity picture of a virus. But to put that in perspective, 82% is also the homology between the mouse and the human genome. So it doesn't take much change to change phenotype significantly. These are amino acid substitutions with frequencies in specific positions of the genome. As you can see, the spike protein is mutating. It's quote unquote looking for better solutions all the time. The RNA-dependent RNA polymerase is mutating less, but this is worrisome because this is the target of remdesivir, and because if it loses fidelity, the rate of mutation is dependent on the rate of error in RNA replication. If RDRP loses fidelity, we're going to see an increase in mutation rate, which unfortunately seems to be happening with the UK variant. Now, this is remarkably similar to what you see in cancer, which is my day job. You see multiple clones of cancer cells that are selected by chemotherapy and replaced by new clones. This is a time course of the appearance of new emerging clades of SARS-CoV-2, starting with 19A, which was the first one in 2019, which is almost gone. And now we have all these new variants that have appeared fairly recently, within the last 2 months, that are increasing in number. In oncology, we call this a tropical fish plot. I'm not sure what we call it in virology, but the phenomenon is essentially natural selection. This is another way of looking at it, a time scale of different variants that have been reported by sequencing sites across the world. There are 19 that have been documented so far, and since the number of genomes sequenced compared to the number of tests run is minuscule, it's likely that there are more. The geographic distribution of these variants is changing. You see South Africa has a very large number of this green variant, which just now has started to appear in the United States. So going forward, just testing people isn't going to be enough. We're going to have to variant call, which is going to require collaboration between multiple institutions in Louisiana and beyond to map the evolution of this virus. The UK variant has been reported in Louisiana, as you all know. The South Africa variant has been reported in South Carolina, and I'm willing to bet a dinner at Commanders that it's already here. Of course, a post-COVID, post-vaccine dinner at Commanders. This is a direct quote from a press release from the LDH today, because most local SARS-CoV-2 laboratory tests are not able to provide results that can identify suspect variant strains. There are likely to be many additional B117, that's the UK variant, cases in regions 1 and 5 that are undetected. So we need to do better to identify variants of concern, for example, in the receptor binding domain of the spike protein or in the RNA polymerase. And we need to improve our downstream data analytics, for example, immunogenicity mapping of the spike protein to determine whether variants can escape immunity. I'm not going to read all the names in this slide. This just goes to tell you that this lemonade took a lot of people to make, from campus leadership to deans and associate deans, to the former Secretary of Health and Healthcare Services Division CEO, Rebecca Gee, who got us the Hyundai donation, 2 departments, genetics and pathology, that actually performed the testing, nurses from the CTRC, ID specialists, the healthcare network, to just give you an idea of how this started. On the first day of testing, and I want to give a shout out to Chris Winters in particular, who's the CEO of the healthcare network and a practicing urologist. We had 14 volunteers who were going to screen. All 14 of them had to be quarantined because one of their teachers was positive. So the day when testing began, it was me and Chris Winters who had to roll up our sleeves and test people for 8 hours. And this went on for a while. So everybody did things that they hadn't done in a while. The School of Public Health that has put in place a good program of contact tracing. Allied health profession, who provided a lot of the volunteers. These are MedTech students. And Ron and San, the BMI core, is analyzing all this data. And all these corporations down here, we paid them well, but they were really helpful because a lot of these things had to come from the four corners of the world because they were not available anywhere near Louisiana. And I'm going to stop here and be happy to take any questions.
Thank you, Lucio. That was tremendous. And thank you for all your efforts, actually, for what you did there. We're open for questions, so we've got a few minutes. Any questions from the floor? I want to make sure I get the one in the chat. Is synthetic vaccines like mRNA vaccine contain bits and pieces that can be readily replaced by others, and spike protein is well conserved? Does this mean that we would be taking a COVID vaccine like the influenza vaccine? This is from Candida Rebello here at Pennington.
So that's a great question. My prediction, and I fervently hope to be wrong, is that we are going to have to take COVID vaccines with some regularity. This virus doesn't mutate as fast as influenza virus because it lacks one of the tricks that influenza virus has, which is swapping segments of genome, because influenza virus has 8 segments in it, so it can exchange pieces with its cousins in chickens and pigs. However, in the last 17 years, 3 different coronaviruses have jumped from animals to humans. And because of the enormous number of people infected, there is a huge reservoir in which the virus can try out all sorts of mutations. So yes, the RNA vaccines can be rapidly adapted. It's estimated it would take about 4 to 6 weeks to change the sequence of a vaccine to incorporate variants. And I anticipate that's going to have to happen.
Very good. Great answer to that. Another quick question came in. We'll take it as from Paul Fidel, but then if you could be brief, Lucio, we've got to move on to Stephania. But do you know how well we're doing with vaccine distribution at LSU HSC?
And actually very well. So what happened is 600 people were vaccinated at Oxnard because these were the people with direct exposure to patients or to samples. Everyone else is being vaccinated on campus. And we vaccinated 1,600 people now in the first week. Now, reservations are dropping a little. So there needs to be messaging. And I've seen in a number of other institutions, refusal rates of the vaccine that are close to 40%. We've vaccinated half of our campus, basically. The other half is moving more slowly. And we're going to have to remind them that this really is necessary.
Yeah. Okay. Very good. Well, again, thank you, Lucio. And I'm going to have to move on to Dr. Cormier. But there are lots of things to discuss there and a lot of very interesting observations that you've made along the way. And I think hopefully a lot of lessons learned for what could be in the next phase of the COVID story. So I'm going to introduce Dr. Stephania Cormier. Dr. Cormier holds the Wiener Endowed Chair and is Professor of Biological Sciences at LSU A&M. She's also Respiratory Immunology and Toxicology Professor at Pennington Biomedical, and has a third title, Honorary Professor in Child Health Research Centre at the University of Queensland, Australia. She is also Associate Vice President of the Office of Research and Economic Development at LSU, and is a world-renowned expert with over 20 years of experience in the field of pulmonary immunology, with a focus on the role of age and the environment in immunity. She has had continuous funding from NIH since 2006, well over $32.5 million in funding. She's director of the LSU Superfund Research Program. This is a large interdisciplinary research center. She is the PI of 2 additional NIH grants, so heavily funded investigator in the respiratory syncytial virus infection domain, asthma, susceptibility to asthma in human infants. And it's a pleasure to have you today. Stephania, title of our talk is Cytokines to Protect COVID-19 Disease Severity. Over to you.
Okay, do you see my screen?
Yep, you're good.
All right, great. So early on during this infection, we developed a role that I really didn't think we would have, but I was sitting in a Gordon Research Conference at the end of February watching COVID-19 start to spread around the world. And when we got back, things changed dramatically and rapidly. My talk is a little bit different than Lucio's. I'm going to focus on some of the data that we started gathering out of this talk, but I'm going to walk you through the process of how we got to that data and where we are now. This is a work in progress. We're still getting some of the patient information, so don't expect a complete scientific story. And feel free to ask any questions. So this is really the timeline of what happened at LSU and Baton Rouge. March 9th was the first presumptive case in Louisiana. March 11th, I was contacted by Dr. Hollis O'Neill. Most of you know him as Bud from the pulmonary group. Bud contacted me, and he had actually run out of viral testing kits. So he didn't have viral transport media. He didn't have swabs. And so they weren't even capable of testing people. So he asked if I had that because I do RSV, which is respiratory syncytial virus. And so we often get nasopharyngeal swabs, and we do certain assays on those to look at infant immune responses to RSV. I told him we don't have those kits. We don't stock a lot of them. We can give them what we had, but we could easily make kits. And so I started investigating how to get these kits. And just like Lucio, I ran into huge supply chain issues. While I could make the media, which was the viral transport media, finding a cotton swab, non-cotton swab, non-wood swab, flock swab, any kind of swab was just not happening. So we had to reach all the way to colleagues across the globe. We reached over to Peter Openshaw, who's actually involved in developing the vaccine in Europe, and we reached over to China, and we had our colleagues there who also do respiratory RSV send us swabs so that we could start making these kits for LSU. We couldn't really make the kits until we actually had we had approval in place for all of these things, and so time is moving rapidly. We're not even a week into this, and we had already written biosafety protocols and started submitting proposals. And by March 18th, the state had come and inspected our lab, and we passed the state sanitation inspection. And so we started creating viral transport media. During this time, we were also getting a biosafety protocol plan in place so that we could actually test for SARS-CoV-2, because we figured that the hospitals, based on information that was coming from the hospitals, were not going to be able to handle the load. And so we started working on that. We started approval for CLIA process March 19th. We had biosafety approvals by the 23rd, and by the 23rd. To give you an idea of the large number of production kits that we did, this is a kit box that contained 50 viral transport media tubes and samples, and those were delivered to the hospitals and to local nursing homes, and even to some of the local state prisons or city prisons for testing. By March 23rd, we had actually already gotten an EUA submitted to the FDA for testing of SARS-CoV-2. And we had, since it was submitted, we were able to start testing on the 23rd for the hospitals. And so we started that night. Now, the bottom line of all that testing is demonstrated here by a model. This model was done by Rebecca Christofferson, who was doing the testing with us. And this was a homegrown effort. So it was her lab and my lab, and we were doing these tests. And the real reason for us doing these tests is the turnaround times for the tests in the hospitals were actually somewhere between, honestly, 4 to 13 days. And so the hospitals couldn't keep up enough PPE, they couldn't supply the rooms with enough staff, and they couldn't move patients from one bed to another, or from a COVID ward out of a COVID ward or to a COVID ward if they needed to be there. And so morale was really low, resource allocation was really high, and it was a tremendous amount of waste. And this model just shows with us coming online how we saved a tremendous amount of PPE. So this is cumulative PPE savings here. And with us operating at only 25% capacity with a turnaround time of less than 1.5 days, we could reduce the amount of PPE dedicated to caring for these patients who actually weren't COVID positive tremendously. Now, as a person who studies respiratory immunology, while I was doing this testing and trying to help the hospitals in my community, the other thing I was thinking about is I study respiratory tract viral infections. And so from our work with respiratory syncytial virus, one of the things we knew is that we could actually look at immune mediators within 24 hours of admission to a hospital and tell you whether or not an infant was going to end up in the intensive care unit or not. And so we applied that approach to our samples from COVID. And so this is just a plot showing you, this is RSV. So this is respiratory syncytial virus. These are infants that were admitted to Le Bonheur Hospital in Memphis. And what we did was take nasal aspirates. And in those nasal aspirates, we were able to quantify cytokines. And then we looked at several other things to see whether or not we could figure out predictors of disease. And basically what this is showing you is these are vectors. And so the length of the vector and the color of the vector demonstrates the contribution of these cytokines or other things like viral load or even gestational age, which isn't pointed out here, but gestational age was also done, and how they contribute to disease severity. And so then we can take that initial high level, unbiased, disciplined look at what's contributing to respiratory disease severity with RSV, and then we can drill that down into specifics, showing that IL-33, IL-1 beta, and then type 2 cytokines were important in driving this response. And honestly, if you keep drilling down, what we could actually drill down to is that if you just knew IL-4 levels on the day the kid was admitted to the hospital, and you knew gestational age in weeks, you could predict with a very high level of accuracy whether or not that kid was going to end up in the ICU. And this was very important for the physicians to know. But we didn't have that. What we actually had was an NP swab. And an NP swab, if you haven't seen what these look like, this is an NP swab, and you can see they go way back here. And as Bud says, they tickle your brain. And they take this out, and this goes into a viral transport media. Now, our viral transport media we made so that we could preserve the proteins in these samples. So it was actually made with things that would inhibit protein degradation and stabilize the proteins in those samples, because we were already planning ahead. Now, what we didn't know, and luckily we were already working on this on the RSV side, is with RSV, we get nasal aspirates from these kids. So basically it's just like taking a pipette, you pipette in about 1 to 3 mils of solution, depending on the age of the child. And then you just collect it. And then from that collection, you can get you can do flow cytometry. You can look at B cells, T cells, you can subphenotype those cells, and then you can look at cytokines. We didn't have that. So with RSV, one of our questions was, since we moved here, it's been harder and harder to get those clinical samples. So the question was whether or not we could use discarded samples to do this. So this is actually us attempting to do this for respiratory syncytial virus, totally unrelated to COVID, but happening at the same time, demonstrating that from an NP swab, we can actually get live cells that we can then stain and then use flow cytometry to identify the subpopulations, along with preserving and getting cytokines from those same samples. So this was basically sort of a proof of concept that we could do this with NP swabs. And so we were saving those NP swabs along the way to see if we could do this with the COVID samples later on. And so for our RTL, which was our River Road testing lab, between March 23rd and May 13th, we ran over 3,857 individual tests. And at the time, these tests weren't multiplexed. So that means something like 12,000 PCR reactions were run by us with a positivity rate of 33%. Now, remember, this was a hospitalized population, so our rate was higher. And the age range of those patients was from 1 to 99 years of age. Now, what we knew is that there were lots of gaps in our knowledge of COVID. Now, we've gotten a lot of information since then, but remember, this is May. And in May, I was working on a grant with Merck to actually look at whether or not we could address some of these critical gaps in our knowledge using the samples that we had collected in the spring, since we had so many. The first question we wanted to know was whether or not disease severity tracked better with viral load or with immune responses by the human host. Now, this was an important question because with RSV, we don't actually see that it tracks with viral load. It actually tracks with the immune response of the host and the age of the host. And then the second question was whether or not immune biomarkers could be predictive of disease severity, sort of like what we saw with RSV. And so to answer those questions, we formed a couple of hypotheses. And those hypotheses was, first of all, that COVID infection initiates a pro-inflammatory response, and then that drives disease severity. And so what we're going to do is use two things: length of stay in the hospital and an Apache 2 score, which is a score of criticalness of the patient, ICU critical score, that can predict actually mortality. And so we're going to use the Apache score. We're going to use primary markers of the inflammatory response and see whether or not it related better to disease severity than viral load. Because while we were running all these samples, we were also running standard curves on each reaction plate to calculate viral loads. In addition, we looked at immune responses, and we questioned whether or not, since we had this huge variability in age from 1 to over 90 years old, our question was whether or not, if we break these down into different age groups, can we look at different age-mediated responses to SARS-CoV-2? And so this was the study we've proposed. And then so most of it's a retrospective study using samples we already have. We take those samples, we're looking at a large number of cytokines in those samples and adipokines and other things that are important in disease, including cardiovascular markers. We're then going to move to also looking at cellular immune responses in those samples. And then we have some prospective studies where we're going to collect serum prospectively throughout an infection protocol and look at cellular immune responses there to see if they differ. Because remember, with our sample, we only have a single time point. So we have the sample when they come into the hospital, which is probably the beginning of peak viral load, but it probably is not the beginning or the peak of the cellular immune response. And so we're just starting to get on that curve of what the cellular immune response is, and it's only a slight window that we're looking at. And what we wanted to do was look at a longitudinal window to see whether or not we could actually look at what's initiating those responses and then what's shutting those down. And we've actually had some case studies come in that have already started filling in these prospective studies. And then we're going to look at antibodies and neutralizing capacity and whether or not those antibodies persisted. So the retrospective study, just to give you an idea of what it was again, we were looking at SARS-CoV-2 positive cases across lifespan. So we limited our study to adults for a number of reasons. One, at the time, we didn't have that many kids. And the disease in kids is going to be very different than adults. So we broke down the adults into 3 age groups: 18 to 59, 60 to 79, and then greater than 80 years of age. And we attempted to grab 50 patients from each of those populations. And then we had a SARS-CoV-2 negative group. These were still admitted to the hospital with symptoms like SARS or indicative of SARS that were greater than 18 years of age. And this was all inclusive of all age groups. And then our prospective study is being done on any adult, and it's going to be a larger group. And they're going to be, these are being enrolled at a local partner hospital. In fact, it's Our Lady of the Lake with confirmed SARS-CoV-2 illness. Okay, so the first thing we did, and so this is a very complex chart, and like I said, we haven't finished analyzing this data, but one of the things we did was we looked at 61 different variables. And this was an unstructured look, meaning we didn't have a hypothesis. And what we were doing was looking at whether or not any of these variables could track with severity. Now, severity, we don't have the Apache scores yet. The our clinical counterpart is still pulling all this data. So for severity, we're using SPO2 and length of stay in the hospital right now. But eventually, this will get modified and incorporate all of that data. But the biggest drivers you can see here, it's not the comorbidities. They seem to overlap between severe and non-severe groups. Other complications, we didn't have enough Hispanics to actually tell anything. Race was actually a good variable. So those two populations separated and were drivers of severity along with sex, so male versus female. And SpO2 levels actually was not a good correlate of severity, which we thought was interesting. Now over here, this is a more complicated plot. This is a plot that shows you the color of the plot actually shows the weight of the variable and its ability to affect severity of disease. And you can see that there are a number of cytokines that are important in this disease. And so we're having what everybody keeps calling the cytokine storm, where a lot of these cytokines are changed between SARS-CoV-2 positive patients and SARS-CoV-2 negative patients. And this is just grouping positive versus negative. This is not across the different age ranges, which we still have to do. But there are a couple of things that I wanted you to note. IL-6 is tracking with positive infection, along with IL-1 alpha, type 1 interferons. IL-29, which is interferon lambda, is important or seems to be tracking very well. And then type 2 cytokines and Th17 type cytokines and BMI. So like I said, we have to actually go through that and then break it down. So what you're looking at on that chart was actually this right here. So we're just looking at positive versus negative for each of those parameters. And here we're looking at these parameters broken down based on positive versus negative, just so you have an idea of what that data looks like. And then across those different age groups, positive versus negative. And so this is just an example. This is the best example. And we didn't even really, I wanted to add interferon lambda, but we didn't really expect a lot of differences. Interferon lambda is very important in antiviral responses. And you can see that indeed all positive, well, the positive patients actually present with a lot more interferon lambda 1 in their nasopharyngeal swabs than those that are negative. In fact, the majority of these are below the limit of detection to begin with. And that this seems to be across all of the different age groups. So no matter what, if you're infected, you seem to have IL-29. And this is interesting because actually there are a couple of, there's a recent report out where they're using pegylated interferon lambda for the treatment of COVID-19 in outpatients. So some of the other things we saw, which I was trying to point out to you on the vector graph, but it's a little bit more complicated, is we saw things involved in Th22 pathways. And IL-22 is actually a cytokine that's involved in lung injury. And it comes up during lung injury, and it's very important in lung repair. And so it makes sense that this would be elevated in SARS-CoV-2 positive patients, as it was. And then cytokines that are responsible for driving IL-22 responses, such as IL-23, are also elevated. And IL-22 shows more of an important role in the younger age group than the older age groups. And it's actually interesting because this sort of suggests that the lack of IL-22 or the lack of the ability to induce IL-22 in these later age ranges may be responsible for the tremendous amount of lung injury and more severe morbidity and mortality associated with SARS-CoV-2 infection. We also saw a lot of cytokines involved in macrophage activation, and that's shown here. MCP2, MCP3 were both elevated in positive patients. And then these also displayed a tremendous amount of activation across the age ranges. So in summary, I wish I had all of this parsed out and I had all the Apache score data and I could break it down even further, but we just don't have that yet. The hospital staff is still pretty busy dealing with COVID. And so we're getting data, it's just not coming fast enough for anybody. But the data will have significant implications, not only for therapeutics like IL-29, which is the interferon lambda that I talked to you about, but also for vaccine development, and even will indicate whether dosing schedules. So this, I think, will come out of our T and B cell analysis work that we're going to do on those nasopharyngeal swabs. And we just don't have that yet because that just takes a little bit more effort. But I'd like to thank Dr. Kirwan for buying the Aurora. This would not have been done without this group of people. So this is our testing team. These people, they work day and night. We all work day and night for nonstop for that entire period of time. On Easter Sunday, I made everybody go home about 10 or 11 that morning. And I said, after that point, I told Bud, I said, we're going to be taking off Sundays from now on. So we worked nonstop until that point. And these kids gave their heart and soul to doing this. And I can't be more proud of any one of them. These are the physicians we worked with, Dr. Bud Hollis O'Neill and Morgan Walker. And then Tanya Jagno came over after she got out of quarantine. And then this is Bev Ogden. She was our medical director, helping us get the CLIA stuff in line. So the people that worked on the data that I showed you, Dr. Luan Vu, who's a postdoc in my lab, Suman Das is a graduate student also working on that, Rebecca Christofferson, and then, like I said, Hollis, Morgan, and Katie is now helping us at Our Lady of the Lake gather this data. And none of this would have been possible except for funding and support through the Baton Rouge Area Foundation and the Health District, and then even private donations. So we did all of this for free. And these are the people who paid for us to do this for these hospitals. And even the hospitals contributed. Some of these people brought us food. Some of these people brought us a PCR machine. So we couldn't have been happier. And then some of the studies are being funded by Merck, a MIST study to study predictors of SARS-CoV-2. And if you have any questions, I'll be happy to answer them.
Thanks, Steph. True scientist, couldn't be happier. Just give me a PCR machine.
Yeah. I have simple needs.
We've got only a couple of minutes left. Are there any questions from the floor? I see a question just popped up from Dr. Ribello.
There is.
Yep. Can you see that yourself? Go ahead.
Okay, so Dr. Ribello is asking whether or not if we can subphenotype the T cells, can we determine whether the immune response will persist or whether the T cells will be able to mount an adequate immune response? So we already know that we didn't know this at the time, but now we know that the T cells do mount an immune response. We get both a CD4 and a CD8 T cell response. What they haven't looked at is strong memory responses. And while T cells are important for the memory response, we're going to look at it and we'll be able to tell you that. But what I'm really interested in looking at is B cells and whether or not we get memory B cells. Because right now we all know that those antibodies are waning. And so the real question is, why are they waning? And so I want to get to that. And if we can answer that and we can figure out how to adjuvant the vaccine better, then maybe we can trigger something that'll create that immunological memory and lead to a persistent response to the vaccine.
Very good. And we have a question from Lucio.
So the Th17 cytokines is correlated with positive for SARS-CoV-2. So Lucio, we don't have those Apache scores yet. And so I can tell you that there are people that have really, really high Th17 responses. And my question that I sent to my student today was, are those people still alive?
Okay.
And I haven't gotten those answers yet because in the IL-17 group, there's these really, really high expressors. And what I'm thinking is the lung injury is so bad that those people probably did not make it, or if they made it, they stayed in the hospital for like a month.
So we actually have, I'd have to talk with our pathology colleagues,
Right.
but we have a number of samples from people who did not make it for sure, because
MP samples?
their autopsies.
Oh, okay. Yes.
In RNA later. From actual tissues of people who died, like lung, heart, brain, and so on and so forth.
Yeah. Yeah, we could take a look at them for sure.
All right.
Right now, I'm trying to get Merck to change the grant. I want them to add this. We originally put it in the grant, and they're like, "Why do you want to know the sequence?" So this is me in May. We need to know the sequence, and so Merck is like, "Take that out, and then we'll fund it." So
Well-
now I'm going back to Merck and saying, you know, I think we should put it back in.
Mhm.
Absolutely.
Yeah.
Great. There are a couple of other quick questions. We are over time, so if you could just maybe quickly address them.
Yep. So Paul Fidel's asking whether or not we can predict sepsis response in positive patients. Yes, I think we can, because we can do it with RSV, Paul. Kayla Fugile, do you think this data will lead to a cytokine screen for positive patients and have them treated? I hope it does. I hope it allows for us to deal with personalized medicine, because as you know, some people are dealing with COVID-19 very well, and then some people get really, really sick.
Mhm.
And so what we want to do is not treat the people who are doing fine on their own. We want to treat the people who are getting really, really sick and prevent them from going to the ICU or getting on a ventilator. So that's my dream, Kayla.
Very good. Good answer. Thank you very much, Stephania. Thank you, Lucio. Great talks today. And thanks to everybody for contributing your questions. Stay tuned 2 weeks from now. I think we have another session. So with that, thank you all and have a good evening.
All right. Take care. Thank you. Bye-bye.
Thank
Hi,
you.
Dr.